Good afternoon, everyone. I'm quite conscious of the fact that the session is running a bit late, so I'll try my utmost to keep to time. And it's great that I'm following up after René. He had an excellent talk about one carbohydrate modification, sulfation. Now I'll talk about a different one, phosphorylation. And what I want to do today is to talk about something that might be a little bit provocative. So a new idea, a new hypothesis, which came to us as a result of a long and winding road down the rabbit hole. So I'm sure that this is perhaps a situation that's familiar to a lot of people here, that we didn't actually have the intention of going out to study phosphorylation of N-glycans in the brain, but you start off in one area reading about topic A, and then you get sidetracked a little bit with references, and then pretty soon you're down the rabbit hole going down topic B, C, D, and you're very far away from where you started. But it's all very interesting. So this was, I would say, curiosity-driven exploration, and driven mainly by our feeling that the more we were reading, there were things that didn't really make sense. And so we had to keep on digging to try to understand this. And I'll say right at the outset that what I'll show here, I'm not going to show any wet lab data. So we haven't done any experiments to empirically test this hypothesis. But what we did do was to analyze and reanalyze existing data sets. So you don't even really often need to do your own experiments, because there's plenty of data out there. We all know with these huge data sets, people just dump a lot of data buried somewhere in the supplementary information. There's a lot there that you can mine. And so that's what we did. And I'll just add that a lot of this originated during the time of the coronavirus pandemic, when we were under various lockdowns and we couldn't work in the lab anyway. So what do you do when you can't work in the lab? Well, you can read, you can write, you can analyze data, and you can think, of course. And that's what we were doing. So this is all kind of what led to this. So I'll take you now on this journey, and present all the evidence, and then at the end of the presentation you can make up your own mind whether this seems plausible or whether maybe it's just nonsense. But we thought we'll put the idea out there, and science will do its thing, and eventually, once this is tested, it'll either be validated or rejected, and that's fine, that's perfect. That's the way science works and how it should. So before we get on to the maybe provocative ideas, we'll start with something not in the least bit provocative, but something very well accepted, and we'll talk about one of the best characterized glycosylation pathways, and probably the example of glycan phosphorylation that's been the most well studied, and that's of course this canonical mannose-6-phosphate phosphorylation pathway, which probably a lot of you are familiar with, and I'll just briefly go over that to recap and refresh for those maybe who aren't. But as I said, extremely well characterized. For decades, people have been researching it. Ajit Varki was working on this pathway in the 1980s. So a lot is known about it. I won't go into every single detail, but just very briefly, we're talking about this modification here. So where a phosphate moiety is added to carbon-6 of a mannose sugar, and this acts as a tag. So its main function is to mark lysosomal enzymes, lysosomal hydrolases, for them to be trafficked to the lysosome. So that happens as these lysosomal enzymes are being processed in the secretory pathway. So it begins in the Golgi, and it's a two-step process actually. In the first step, these enzymes, which have usually oligomannose N-glycans somewhere on them, and hence they have these free terminal mannoses available. This enzyme called glucose phosphotransferase transfers a phosphate group to the mannose, but it also transfers attached to the phosphate this glucose. So in this first step, that mannose is actually masked. It's not active. It needs to be unmasked for it to be recognized by its cognate receptor. So that happens in the next step by something called the uncovering enzyme, which trims that glucnug, and then you finally have your free and available phosphate group, which can be recognized by a specific receptor called the mannose-6-phosphate receptor, logically, and that transports those enzymes to the lysosome. And they're released in the low pH environment, and they can do their business of degrading various biomolecules. So, needless to say, this is a very important pathway. It's critical to cell functioning, and any disturbance of this pathway can lead to disease. Most notably, we're talking about lysosomal storage disorders, where if these enzymes are not getting to the lysosome, then of course you get an accumulation of their substrates which are not being degraded. So that causes a big problem, of course. And a few examples of those are these mucolipidosis disorders, 2 and 3. So that's in a nutshell what's known about that pathway. Just to add to that, the topology and the number of phosphates added to these glycans determines their binding to the receptor, determines their affinity. So the more phosphates you have, of course, the better the affinity, but also the position. And through many decades of research, it's known where they can be attached. So, of course, it's predominantly oligomannose and glycans, can be hybrid as well, because they have free mannoses. But here I've just drawn on a representative mannose 9 precursor molecule. In dark green are all the phosphates that can be phosphorylated, all the mannoses that can be phosphorylated, pardon me. So basically all of them except for the 3 core mannoses. However, there are some that are preferred, and they're marked with these asterisks. So what do we know about phosphoglycoproteins in the brain? Well, one thing we know is that they are especially rich in these phosphorylated oligosaccharides. They contain almost double the amount of the next tissue, which is the testis. And this is not due to a higher concentration of these enzymes in the lysosomes, nor is it due to their accumulation in other organelles. So that's been ruled out. So the current thinking is that actually, for some reason, in the brain, the removal of this tag works a bit differently, and it's less efficient than other tissues, which results in an extended half-life of this mannose-6-phosphate tag. But which glycoproteins bear these phosphorylated N-glycans? And the answer to that question, as Yuri mentioned yesterday, it can be a trivial thing, but it's worth emphasizing that the answer to that question depends on which method you use. Of course, any method that you use has its limitations, and it's like looking through a keyhole. You'll only see what you can see with that particular keyhole, right? Not the whole scene, just the part. And in the early days, decades ago, when people were researching this pathway, of course, the primary tool that people used was to use that receptor I mentioned, the mannose-6-phosphate receptor, which that was purified, and you could then use it as a tool to fish out phosphorylated glycans and phosphorylated glycoproteins. And that was great, it works very well, and most of the studies used that at the time. Of course, you're using a receptor, which its function is to transport lysosomal proteins. So of course, it's not really a surprise that you're probably skewing your view of things towards the lysosomal enzymes. Okay, so here I've made a Venn diagram showing how many and which different glycoproteins have been detected in the rodent brain in 3 different studies, and using 3 different methods. And the shading shows the proportion of those in that sector that are lysosomal enzymes. So no surprise here, they're majority lysosomal. But you can see with these 2 other methods, and they're more recent methods, so of course methods develop and people invent new methods. So these are what I would call unbiased methods. Well, actually this uses concanavalin A, which fishes out oligomannose N-glycans, but of course they're the ones that are mainly phosphorylated, so that's fine. But this method by Huang et al. Uses something called IMAX, so it's immobilized metal affinity chromatography. So the positive charge on the metal, in this case titanium, attracts the negative charge on the phosphates. So it's purely a charge-based purification. And of course, no surprise, they see different things. And then I've highlighted just in this sector the 6 glycoproteins identified in 2 different studies, which are not lysosomal, none of them. But, you know, the degree of confidence is quite high that they contain phosphorylated N-glycans, and they are not lysosomal. So separately identified in 2 different studies using two different methods. And when you look at all these glycoproteins, you can group them into several different categories. So the first group is those that are known lysosomal enzymes, and they're recognized by the mannose-6-phosphate receptor. So that's all good and logical, and they're all here. Then you have some that are lysosomal but not recognized by the mannose-6-phosphate receptor. Okay, there are a few exceptions. We know that that system is not 100%, so you do get some lysosomal enzymes somehow getting to the lysosome without this tag, so there are clearly backup pathways or alternative pathways. And vice versa, some glycoproteins get the tag, but they don't end up in the lysosome. Of course, biology is very complicated. So on the other side, vice versa, you have the other situation, and that's okay, we've got a few exceptions. But then you have this interesting group, which are known not to be lysosomal enzymes, and they're not recognized by the mannose-6-phosphate receptor, but they are phosphorylated. I mean, they do carry phosphorylated N-glycans. So my question, logically looking at that, was why are so many non-lysosomal glycoproteins being modified by phosphorylated N-glycans in the brain? And what is the function of this phosphate modification if it's not to transport them to the lysosome, which is what mannose-6-phosphate does, because they're not located in the lysosome. A lot of the data I'm about to show that follows involves mining the data that was generated here, this data set from Huang et al., which was a very rich source of data. So they published this paper in 2019 where they developed a new method, or I should say they refined an existing method for enrichment of phosphorylated N-glycans. Based on this IMAC that I mentioned, they just added, so that was already existing, they added now a sequence of washes through gradually stepping down the ACN concentration. Anyway, I won't really go into too much detail. I do like that fountain analogy, though, with the cascading washes. So it was really a methodological paper, and they then tested the method in a few different tissues, and they found that their method that they optimized was superior to old methods in enrichment of these types of phosphorylated N-glycans, and they had all this data. But that's kind of where they left it. They didn't really go into any biology. So I, on the other hand, was very interested to see what kind of, because they had the glycopeptide data, what kind of glycoproteins are phosphorylated in the brain. So we mined that data, and the first thing that we saw was that not only is the brain rich in these phosphoglycoproteins, it also contains an abundance of brain-specific phosphoglycoproteins. So more than any other tissue that they looked at. So these ones in gray, the ones that are shared across multiple tissues, they're likely to be lysosomes, of course, lysosomal enzymes, because they're more or less ubiquitous. But this is the interesting part. Almost half are specific to the brain. So, another question that arose out of this was, are there any differences in the way that lysosomal and non-lysosomal glycoproteins are modified by phosphorylated oligosaccharides? So taking out all of the glycoproteins identified in the mouse brain that contain these phosphorylated N-glycans, we split them according to the subcellular localization into 2 groups, lysosomal and non-lysosomal. There are a couple here in gray. So blue is lysosomal, pink is non-lysosomal. Gray ones are, the literature is not clear, and sometimes they shuttle or they exist in multiple. So we completely ignored those that are variable, just to make things clean. But hopefully looking at this, so here we've mapped the N-glycosides where the phosphorylated N-glycans occur. And hopefully what can be immediately visible just from this overview is that there are definitely differences in the way that lysosomal and non-lysosomal glycoproteins of the brain are modified. So one thing is that if you just look at the number of glycosides that contain these phosphorylated N-glycans, there are much less in these non-lysosomal glycoproteins. In fact, maximum of 1 per protein, whereas lysosomal enzymes can have up to 3. Then you look at this microheterogeneity. So how many different kinds of phosphorylated N-glycans occur on those sites? And you can see here it's scant, 1 or 2 max, whereas that's for the non-lysosomal. Whereas the lysosomal ones can have many more of these, so a greater microheterogeneity of these kinds of phosphorylated N-glycans, and that's statistically different between those groups. So could this be, though, just to a general decrease in microheterogeneity in the non-lysosomal ones? It's a fair question. But to analyze that, we would need to look at phospho N-glycans, but in the context of the whole N-glycome. Because in this paper that I mentioned, Huang et al., they actually only focused on phosphorylated N-glycans and discarded all the other data. So we lost that bigger picture. But of course, there are other studies. So we used this excellent paper, also published 2019, using a different method, where they did total N-glycopeptide profiling of the mouse brain, including, of course, phosphorylated N-glycans, which are these yellow lines here. So now we have the whole context, and we can look at the N-glycosylation sites that have these phosphorylated N-glycans, but in the context of the whole N-glycome. And that's the figure I showed before. But then we ask the question, okay, the microheterogeneity is less in the non-lysosomal ones, specifically for phosphorylated N-glycans. But what about total microheterogeneity? Well, no real difference. But okay, phosphate goes on oligomannose N-glycans. Maybe there's just a difference in glycosylation or microheterogeneity in specifically oligomannose between the groups. Again, no difference. So what is that telling us? Well, it suggests to me that this difference occurs at the level of phosphorylation, not at the level of glycosylation. And we went deeper, and we wanted to then actually ask, well, is there something about the peptide sequence, or the protein sequence in the immediate vicinity of these N-glycosylation sites that carry phosphorylated glycans that can explain this, that leads to a difference? You know, is there maybe a motif, an amino acid motif in either lysosomal enzymes or non-lysosomal ones, which could explain this? But no, when we looked 25 amino acids upstream and downstream, we didn't find any enriched motif in either group. So the conclusion is that there must be factors other than adjacent primary protein structure that affect phospho and glycan microheterogeneity. So winding up now, we're almost at the end. Just another thing we wanted to then look at was the distribution of different phospho and glycan compositions according to the protein type, lysosomal or non-lysosomal enzymes. So here we have all the ones that were detected in the mouse brain and their distribution. So this is the most common form. It's this M6 oligomannose and glycan with one phosphate. You sometimes get diphosphorylated ones. So you can see there's a difference between lysosomal in blue and non-lysosomal in pink. But what was really interesting for me was that actually you get some that are unique or some that are specific. In other words, these ones marked in blue. So these phospho and glycan compositions only occur on lysosomal enzymes, and this one. And vice versa, these ones in red never occur on lysosomal ones, but can occur on non-lysosomal glycoproteins. So I don't know what it means exactly, but it seems to me that there's something happening here, that there's a difference in the catalog of phosphorylated glycans present in these two classes of proteins. And in fact, going back to what I said at the beginning of the talk, this is just the core N-glycan structure with one phosphate, detected in this study, but also detected in numerous others in the brain. So I know about 4 or 5 papers, including we've detected this in our research in the brain, that this should not be possible, actually, according to what we know about the mannose-6-phosphate pathway. That's never been described for mannose-6-phosphate, that the phosphate goes on one of these mannoses. Is that just an experimental artifact? Who knows? But it's an interesting observation that you don't see it on lysosomal ones, but you do see it on other kinds of proteins. And now maybe, for me, the most tantalizing piece of evidence, which comes from an ancient study, a very old study. So it's tantalizing because it's potentially the strongest evidence in favor that there's maybe something different happening in the brain and a different kind of phosphorylation pathway. On the one hand, yes, but on the other hand, it's very old. It's an old study, and it has not been replicated in the almost half century since its publication, unfortunately, as far as I know, to my knowledge. Please, if someone else knows of a more recent one, I would love to read it. But there was this study from a very reputable lab, so from a very respected and a top researcher. It came from the Brungraber lab. So, if it was another author or another lab, I might be more skeptical, but I've read many papers from that group, and all of them are, top quality, rigorous, etc. So I'm more inclined to take it at face value. But anyway, they did a very interesting study where they used NMR, and they had evidence from studies on rat brain proteins that in the brain, phosphate groups can be attached to other carbon atoms, so other carbon positions in the mannose residues. For example, the 2, 3, or 4 carbon, which is highly unusual, but definitely is not in keeping with the mannose 6-phosphate pathway, because it's called the mannose 6-phosphate pathway. It's in the name of the pathway. So that would be a departure from that. Anyway, that still remains to be verified. So, like I said, it's old, and I would love to. If anyone does NMR here, please talk. I know, Robert, you mentioned NMR, so maybe we can chat later. But how are we going for time? I think we're on time. I'll just summarize. So, individually, when you look at any one piece of this evidence individually, it's not particularly convincing or strong. But the point I'm trying to make here is, if you take it all together and combine it into, look at the whole picture, I don't know, to me there's something fishy going on, and to me this suggests that there may be fundamental mechanistic differences in the way that N-glycans of lysosomal glycoproteins are phosphorylated compared to those of non-lysosomal glycoproteins. And it's clear, if nothing else, that these phosphorylated N-glycans in the brain exhibit some distinctive features that don't completely align with what we already know about the canonical mannose 6-phosphate pathway. So what are the options? How can we explain this? Well, one option is, to question the canon, but that's almost sacrilegious. And of course, this evidence is nowhere near strong enough to make such claims yet. But could there be an alternative possibility? Both things could be true. Maybe everything we know about mannose 6-phosphate pathway stands, but perhaps in the brain, could it be that there might be an additional alternative oligosaccharide phosphorylation pathway that may be active in parallel, and it may be modifying a discrete set of brain glycoproteins, and it has a completely different function than mannose 6-phosphate. This is just a summary table basically showing what I had on the previous slide, but I think time is up, so I'll stop here and acknowledge all my co-authors who were involved in this, and I'll be happy to take questions if time, but I know we're running late, so the chair will dictate.
Thank you so much.